Almost 30 million people in the United States were diagnosed with alcohol use disorder, or AUD, in the past year. That's more than all other substance use disorders combined. AUD has a significant negative health consequences and can ultimately be fatal. And while many people drink, it's individuals who drink to high levels of intoxication that are at greatest risk for AUD. And Illinois is not doing so well. We have some of the highest rates of binge drinking in the country. At the national level, the incidence of AUD is high, but decreasing. But we've seen a resurgence in AUD rates in Illinois and Cook County in recent years, with some rates far exceeding the national average in distinct Chicago neighborhoods. So this is a big problem nationally, but arguably an even greater problem for us locally. And it really speaks to the need for us to better understand why people drink and how we can help them to stop. Now, addiction is a brain disease, and my lab is combining breakthrough technologies in neuroscience with a rat model of AUD to try to answer these questions by understanding how brain cells govern AUD symptoms. For example, what I'm showing you here is how we combine lasers and fiber optics to measure calcium in the brain. This allows us to correlate brain cell activity with behavior. We use techniques like opto and chemogenetics, which deliver light or chemicals to the brain and allow us to turn individual brain cells on or off with a fantastic level of precision. For example, here, we're turning on a set of cells that facilitate movement in an animal that isn't moving. Now, using these approaches, my lab identified a population of cells in the brain that are hyperactive during withdrawal at a time when our animal model, just like people with AUD, experiences heightened pain sensitivity. You can see that here with alcohol-dependent animals withdrawing more quickly from a painful heat source compared to controls. But if we turn these brain cells off, we can essentially rewire the brain to reduce pain, thereby reducing the drive to drink. Now, these approaches are really useful tools for understanding how the brain works, or doesn't in the case of AUD. But they're probably not the approaches that are going to be used in the clinic anytime soon. What's exciting, though, is that in collaboration with UIC's Center for Alcohol Research and Epigenetics and the Institute for Genomic Biology at UIUC, my lab is combining the approaches I just told you about with experiments that allow us to look at how DNA is organized within the cell, or epigenetics, and how that leads to changes in gene expression. And we're now linking these changes with AUD symptoms. Even more exciting, with access to an AUD brain bank, we're able to translate the findings we uncover in our rodent model into humans that had AUD. And it's these approaches that I'm the most excited about because they hold the greatest promise for uncovering unique molecular targets for treatment development. Thank you.